Hello, and welcome to this webinar titled Achieving Seamless Scale-Up and Technology Transfer, a Case Study in Single-Use Bioreactors, hosted by Biopharma Asia magazine and presented by Ying Wang, Ph.D, Senior Scientist of Manufacturing Sciences at AbbVie Bioresearch Center. My name is Stephen Edwards, and I'll be your moderator. Now, please allow me to introduce our presenter. Ying Wang is currently a Senior Scientist in the Manufacturing Sciences Department at AbbVie Bioresearch Center in Worcester, Massachusetts, USA. He got his bachelor's and master's degree in chemical engineering at Tsinghua University, China. He got his Ph.D. in chemical and biomolecular engineering at Johns Hopkins University, USA, and joined AbbVie in 2015. His research focuses on the development of bioprocesses based on biological understandings and engineering methods, using mammalian cells for producing recombinant proteins and monoclonal antibodies. His expertise in biologics production from Chinese hamster ovaries or CHO cell cultures, especially for upstream process development and scale-up. He had experience in multiple commercial and late clinical stage biologic products, including MABs and fusion proteins, performing technology transfer, cell culture process validation, scale-down model qualification, bioreactor KLA studies, and driving innovative study in metabolic pathways. He also had experience on authoring sections and providing technical support for BLA filing, regulatory audits, and response to the comments from agencies. He has also had research experience during his Ph.D. study, which focused on human-induced pluripotent stem cells, gene editing, stem cell differentiation for translational medicine. I'll now be handing over to our presenter. Welcome, Dr. Wang.
Thank you very much, Stephen. Good morning, everyone. This is Ying Wang. I'm a senior scientist in Manufacturing Sciences Department at AbbVie Bioresearch Center. Today, I would like to talk about how to achieving seamless scale-up and technology transfer for CHO cell culture process. This is going to be a case study in using single-use bioreactors. So first of all, I'd like to give a quick introduction of AbbVie Bioresearch Center. So we are located at Worcester, Massachusetts in the United States, and the major duty for our ABC Bioresearch Center is the biologics discovery and development, and also manufacturing. And this is a unique site that combines both R&D and manufacturing together. So we are in charge with the clinical and commercial manufacturing of both AbbVie and third-party products. We have over 23 years experience of handling over 100 different biologics so far. And we have a big GMP facility, a manufacturing facility. We have two 3,000-liter production bioreactors in two independent suites. We also have 5 6,000-liter bioreactors in another two suites, and two 1,000-liter bioreactors in an all single-use suite. And we have 5 independent purification suite and the Class B bulk fill suite. So this is the AbbVie Bioresearch Center. And you can see probably from online that AbbVie have a very robust pipeline for biologics and in a wide range across oncology, immunology, and neurosciences. And normally such a big pipeline and also plus some accelerated filing strategy requires a very systematic scale-up, scale-down, and technology transfer strategy to facilitate this. So the benefits for a good tech transfer strategy, it can rationalize the process design, it can streamline tech transfer, and also platform scale-up, scale-down to save time. And today's presentation will be covering the technology transfer and scale up with some general ideas and followed by a detailed case study. So overall, there are several key stages in late phase process for tech transfer. First of all, we need to consider a process fit, which basically you need to transfer from one site to another. You need to identify gaps and mitigate the risk that can potentially happen during the tech transfer. And since it's late-stage process, it's normally we want to target to make minimum process changes as possible. Then the second part is going to be the tech transfer and scale change. Normally, it includes a lot of key tech transfer documentations and studies beforehand. For example, we need to set up a master plan. We need to have a lot of risk assessment and process failure mode and effect analysis. For cell culture part, we need to do some KLA studies, mixing studies to understand the equipment, and we need to establish comparability protocol and PPQ protocols. And then once everything is ready, we need to execute the PPQ runs. PPQ is kind of a very important milestone, a process performance qualification. The target here is to meet all the in-process control limits, specifications, and product quality requirements. And also another important thing to show to demonstrate the comparability between the current site or process to the previous process. So these are the key elements. In terms of cell culture process transfer or scale change, we are facing some of the very important parameters, such as the agitation rate, pH control strategy, DO set point, airflow cap, oxygen flow capacity, gassing strategy, and feeding strategy, and so on. These are the process parameters we need to determine when we transfer a process to another site. There are some of the parameters that are easy to transfer, such as the pH set point or DO set point, which are scale independent. But then there are some other process parameters which are more important or more difficult to transfer because it depends on the reactor you're using, and such as the agitation rate or the airflow cap or the gassing strategy. So all these scale-dependent parameters, we need to rely on some non-scale related parameters to make the change, such as considering the similarity of bioreactors geometry, because if you transfer a process from a reactor to another very similar reactor, it's going to be a lot easier. And we need to rely on some, for example, mixing time and shear stress estimation, and some other non-scale related parameters, such as the volumetric mass transfer coefficient, the KLA, which we can talk a lot about it today, and also power input, which kind of an estimation of how much power or mechanic damage that transfer from the impeller, the agitator, to the cell culture, and also superficial gas velocity and volumetric gas flow rate. Those are all the kind of key parameters that we consider during cell culture tech transfer. And in terms of the case study, I will show you an example how we do or determine all these parameters. So this is a task when we facing. It's going to be a tech transfer from a phase 2 200-liter scale from site A to a 1,000-liter manufacturing scale, commercial manufacturing scale in site B. It's a big change and a big scale change. So the challenge we're facing is majorly process feed, and we did need to plan some process changes to accommodate this. So one big thing is the bioreactor system are different. So in the 200-liter scale is using one type of single-use bioreactor. In the 1,000-liter, it's a different system. And then the gassing strategy alongside is because of the bioreactor difference. So for the 200-liter, we're using microsparger to control the oxygen, and use drilled hole sparger to piping air to control CO2. And then in the 1,000-liter, we use one set of drilled hole sparger for all the air, CO2, and oxygen flow. So this is a big difference that we need to consider. And then the pH control logic between the two bioreactor systems are different. The 200 liter using a deadband control, which basically you can think to control the pH in a highway that you can drive as long as you drive between the curbs, it's fine. It won't have any control. And then the 1,000 liter pH control logic is the set point control, which more or less like a train on the trail. So you need to control it at their set point all the time. And then the harvest strategy is also different. 200 liter has a cell settling step, and followed by depth filtration, and the 1,000 liter we want to only have depth filtration without the cell settling. That takes for over a day. So this is the challenge we're facing. There are major process changes need to be made. On top of that, we have a very tight timeline. This is the oncology project, and there are some aggressive filing strategies. So it's basically from the beginning, from the first technical team telecom to the first saw of the PPQ campaign, we only have 18 weeks. So how to manage this without even have a chance to have engineering around? So here is our strategy we used for the tech transfer, for the cell culture strategy. So first of all, we want to establish a scale-down model in the lab to enable any studies that we can encounter, and also kind of have a proof of concept in the small scale to see whether our plans changes make sense or not. And then the second step is to scale up to a 200 liter pilot scale, because we want to transfer to the same single-use bioreactor system, and also test all the plan changes, especially for the gassing, so that we have some more confidence to scale up to just 1,000 liter. And then the other small task we added in the 200 liter is for extended cell age PPCB generation. And then the next step is going to be, of course, scale up to 1,000 liter scale and be ready for the PPQ. The goal for each step is the same. Basically, we want to chase for comparable cell culture performance and also, most importantly, product quality. And then we basically break down this timeline for acceleration project just to showcase. So first, you can see we have a team meeting call, and then we saw the first vial basically a week later, and then we have some other face-to-face meeting. And when we complete 3 rounds of scaled-down model development study in the lab, and then we complete the KLA study at the large scale, we write the process flow diagram on week 10, and then 200 liter pilot run after nearly 2.5 months, and then scale up upstream batch record draft for the PPQ round. To achieve all this, original strategy normally we do is we set up a master plan, and we do a pre-calculation risk assessment to adjust what's the important parameters we need to study, and then we do a process calculation which studies every individual important process parameters and make sure what range we can operate within, and then we do a criticality assessment to establish all the control ranges for the PPQ. But in this case, we don't have the time or luxury to do all the process calculation studies at the same time. So basically, we took more risk assessment based strategy. We moved the process calculation and criticality assessment alongside the PPQ process. And then we established basically QA risk assessment, control strategy, and some PJR, PPQ protocol, comparability protocol after all the risk assessment. So the key for this, to achieve all this tight timeline, is to have a very well knowledge transfer and have close communication. We need to make sure whoever taking charge with the responsibilities, and then we need to perform all the tasks in parallel. And of course, we need a systematic scale-up strategy. So first of all, we do the 3 rounds of scale-down model development. As you can see, it's also a very big change. We need to change the sparger and gassing strategy, as I mentioned before. So first of all, we tried the microsparger in a 3-liter bioreactors, but the result didn't turn out well because it shows us reduced cell density, and it also shows lower titer. The reason for that is in the microsparger, it's very difficult to control the DO at the set point, and it generates a lot of foaming. But then we found that after we switch all to the drilled hole sparger as we normally use, it shows very comparable titer and cell growth. So we have a steady DO control using the drill hole sparger, and oxygen flow requirement, of course, is a lot higher because it's the gas transfer efficiency is not as good as the microsparger, but we do see robust growth, titer, and comparable product quality. And then the second big change is the pH control logic. As we said, here we tend to use set point control because it's easy for process characterization and easy for a more robust and reproducible control. So we want to change it from deadband to a set point control. And you can see here the black line is the phase 2 200 liters process. The pH is free falling and then controlled within a band. We managed to repeat the deadband logic in our 3-liter bioreactors, and we also kind of change it finally to the green line, as you can see here in the 3-liter as a set point control, with a pH shift around in the middle of the culture. And we've noticed that the process performance was all comparable across the board with different control logic. So the tighter pH control is definitely going to improve the overall consistency, and it did not result in a higher base usage. So we are comfortable with the switch for the pH control logic. And then the next step is to how to scale up all these changes to a larger bioreactor. Of course, the focus is going to be several folds. One is to make sure that there's not large or big mechanical damage, and we need to make sure there are acceptable shear stress from the agitation or from the gassing to the cell culture. And then the second one is we need to supply sufficient oxygen, right? So it's also related to mass transfer and mixing. And the third point is similar microenvironment. Normally we mean PCO2, the carbon dioxide environment for cell culture. So there are 3 steps we take. First one is to determine the agitation rate, because once this is fixed, others will plan or study around it. So first step to determine the agitation rate in the larger reactor. Normally, traditionally, people can calculate the impeller tip speed just to make sure it is larger scale is smaller or equals to the smaller scale. And then later on, for what we were using was the impeller power input to match the impeller power input, which are normally used across the industry. And also we do computational simulation to see how the shear stress can impact the cell culture. And also we're considering the mixing time and KLA oxygen with the max for the mass transfer part. And then once the agitation set point is set, in step 2, we will need to make sure that the oxygen supply is sufficient in the new bioreactor system, since we changed to the larger the drill hole sparger. This process is also known to be a high oxygen consumption, high oxygen consuming process. So this is a big concern for us. What we did. Traditionally, people will comparing the VVM or volumetric oxygen flow at the peak density to see whether the new process is capable. And then what we did was to compare the KLA oxygen at the peak density with try to make it have a comfortable margin, assuming that we can reach the similar peak density and the cell has similar oxygen uptake rate. And then the third step is determining airflow cap. Since we provide oxygen in such a way that we will supply air first to strip out CO2 as much as possible, and also supply oxygen at the beginning of the culture. And then later on, since air is not enough, we need to supply pure oxygen. So airflow cap is a way set the process parameter that we control. We try to control the CO2 level in the culture. So we also took a KLA-related, KLA-based strategy. We basically, normally people will do to match the volumetric airflow cap across the different scale of the bioreactor. What we did is we tried to match the KLA CO2 at the airflow cap. I'll put more details in this later. But overall, this is our strategy from 3 steps to scale from a smaller bioreactor to large bioreactor. So you can see here, this is the detail. First, we determined agitation rate. We did all the calculations for power input and relevant accumulative shear stress by theory. And then we do some computational modeling just to see what the shear rate distribution in the bioreactors. You can see the 1,000 liter, the shear rate ranges is even smaller than 3 liter bioreactors, which is satisfying. And then we also calculate the micro mixing time based on the simulation, and we don't think the large scale bioreactor need a long time for mixing. It's a 1,000 liter scale. Anyway, it won't be a big trouble in theory. So in this way, we determine the ideal agitation rate by matching the power input number. And next step is the KLA study. As I mentioned, KLA is volumetric mass transfer coefficient. It describes the rate of gas transfer from the liquid phase to gas phase, or the other way, from gas phase to liquid phase. And it's controlled by several factors. It's the geometry of the bioreactor, sparger design, impeller design. These are not changeable once we have the reactor set, but we can change the agitation rate and gas flow rate to control this. So the general way is to use experimental data to establish multiple points, and then we feed to a calculated correlation with the consistent A, alpha, and beta as the equation showed below, and we can establish the KLA in a wider range to be built in the model. So the way we measure the KLA is a gassing out method. So basically, we're measuring KLA oxygen and KLA CO2 spontaneously. The overall method is that at the beginning of one conditions, we will pumping nitrogen and CO2 together to both bring the dissolved oxygen level down and also keep a pH at a certain level. And then we turn off the nitrogen CO2 and turn on the airflow at a certain agitation rate and airflow rate. So the air will supply oxygen in the solution. You can see the DO is increasing, and also at the same time strip out the KLA CO2, strip out the CO2 from the liquid, so the pH is dropping. So then later on we can fit into curves to calculate the KLA for both of the numbers at the same time. And then, so to confirm the oxygen transfer capacity, we take this KLA-based approach. Basically, we are first understand what the maximum oxygen demand based on the smaller scale runs at the peak oxygen flow, and then we collect all the KLA oxygen at different reactors and then we calculate whether the 1,000 liter mass flow controller can provide this high of KLA. And here is what we do normally. So for the 3 liter, we would know what the agitation, what the peak oxygen flow is, and we we think, oh, this is going to be the peak KLA CO2, peak KLA oxygen we need. And also, at the same time, we did another KLA confirmation using a cell culture method. Basically, you turn air and oxygen during the culture and calculate how fast the cells consume that, and then supply back air and oxygen to calculate how the KLA is, which we did confirm that it is A1, let's say, for a number. And then using this number, we also confirmed the 200 liter has a very similar KLA at the peak density. And then the 1,000 liter, we should match at least A1. And then using 1,000 liter KLA information, we found that here is the maximum oxygen flow rate that the bioreactor can supply, and A1 is here. So we reached the conclusion that whatever the maximum capacity we can supply in the bioreactor can match the cell culture demand. And then the similar thing for the KLA CO2 is that when we set the airflow cap of the 3 liter bioreactor, we have a KLA CO2 here, and then we set the same airflow cap to reach the same KLA CO2 to make sure that at least at the airflow cap, we are stripping the CO2 at the same rate, so we can have similar PCO2 in the culture. This method worked best in a low oxygen flow culture because their entire culture is more rely on air, so it's sufficient to match PCO2 profile at the majority of the culture. But in a higher oxygen flow rate, you probably need to take additional actions to adjust the agitation later on, especially in a small scale model, because it's normally stripping too much, and it's hard to mimic the actual PCO2 profile in a larger bioreactor. And here are the results for the final process transfer. So you can see the black line is the purple line is the phase 2 200 liter result. We achieved slightly higher peak VCD, and then the viability is comparable. We are still within the harvest range, and we have seen especially for the 1,000 liter bioreactors, we achieved slightly higher titer compared to the phase 2 process. And here are just 2 examples of the critical quality attributes, which is in line with the phase 2 200 liter result. And so our conclusion is that we have a very comparable cell culture performance, comparable titer and product quality, and we did all this and enabled the PBQ within 18 weeks. As a summary, we can see a systematic scale-up, scale-down strategy is very useful because it can rationalize the process design, which is very helpful for the filing later on, and then streamline the tech transfer, and then we have its platform to scale up and scale down process to save time. And the strategies, we took a method to match the power input calculation and the CFD modeling for share to determine the impeller type and agitation rate, basically. And then the KLA-based strategy give us advantage to confirm the oxygen transfer capacity and also rationally set the airflow cap to match the PCO2 profile. And we think using this strategy achieved prompt and successful tech transfer and scale up in this case study, and we achieved the comparable cell culture performance, titer, and PQ in a very short time period. So, of course, it's a very collaborative effort from multiple departments. So these are colleagues I would like to give acknowledgement. And that's it. I'm welcoming all the questions.
Thank you, Dr. Wang. As you just mentioned, now, yes, we will begin the question and answer segment of the webinar. But once again, I would like to remind our audience that you can still send your questions in via the Questions tab located directly below your webinar screen. So now, our first question. Is matching KLa CO2 at airflow cap sufficient to achieve comparable pCO2 for the entire culture?
So in this case, actually, we are not quite satisfied for the KLa CO2, just matching the KLa CO2 at the airflow cap. The reason why is that since this culture is very high oxygen demand, so it's only can cover the first 3 days. But normally, in terms of the impact, this is sufficient since we know that higher pCO2 can impact the cell growth from early on, and you want to control it later. Once the pH control switch to CO2 mode later on, it doesn't matter much. But once you start to see your lactate consumption, your pH going up, you probably want will add base toward the end of culture. That's why we need to make some adjustments with agitation or pH set point to keep the pCO2 in check. But overall, this is worked fine in this case, and it also works well with a lot better in some other low oxygen demand culture. So I think this is sufficient for now, and there's more adjustments need to be done.
Thank you. Our next question is, without process characterization, how are the normal operating ranges for the process parameters determined for the PPQ?
That one I think we do have previous experience from the other party of the tech transfer, so we do have some process understanding, also based on some platform knowledge that we have been doing. And normally, as for now, we are more lean to set it tightly at the beginning, especially for the normal operation range, based on the equipment capability or calibration capability. So that's normally acceptable. That's normally acceptable by the agency based on our experience.
Thank you. Our next question. What do you do if the pH strategy change does not show comparable results?
I guess we've seen that in other process as well. When we're trying to switch from deadband control to set point control, it not always show as comparable. What we did there was we did try to match back to the deadband control. So we also sometimes tried one-sided control, switching from CO2 mode to base mode to prevent additional base addition. But we do normally try to see whether if we switch to deadband control, what's going to happen. In this case, you can see we still can do deadband control in the 3 liters, but I think keep both options will be good. Yep.
Thank you. Now moving on to our next question. Do you try to mimic high mixing times of the large scale in the small scale model?
No, we didn't. This is difficult. I mean, in this particular case study, we don't. But then in other process, we did see some uncertainty about what's the scale impact on the culture. Say if we see a product quality impact when we scale up in a larger reactors. This is 1,000 liters, so it's not quite big, but sometimes we scale up to a 6,000 liter or 12K. We do see that. We did try some, say, for example, low agitation or delayed pH control or DH control, or some like culture exchange type of lab setups to mimic the high mixing time. But overall, in this case, we didn't, but we do try this effort in the lab.
And thank you. Our next question: what kinds of PCO2 levels do you try to mimic in the 3L system, and how do you do it given the high stripping capacity?
Yeah, this is a very good question. Overall, it is a kind of a universal problem for a 3-liter system, especially nowadays. People do a lot of high-density culture, and this stripping in the 3-liter system is massive that we basically couldn't even get close to the larger scale. So in 3-liter, in this case, we basically, overall, we are thinking that as long as we are not exceed a certain level of PCO2, it should be fine. And we do try, in some cases, try to basically manually add a little base or manually let the pH control fight each other to kind of intentionally bring the PCO2 level higher in the 3-liter bioreactors, and also try to, like, say, reduce the airflow cap or something. We did try that. In this case, we didn't see a big impact, but that's something that we should do when we do process characterization in a 3-liter. Some effort we should put in there. Yeah, hope that answers the question.
Yeah. Well, thank you for your answers, and thank you, audience members, for your questions. Before I finish the webinar, I'd like to ask our presenter, Dr. Wang, if you have any closing remarks.
No, I think thank you everyone for listening, and hope the webinar is helpful for your process development. Thank you.
Well, thank you for that. And I would like to take this opportunity to thank our presenter. Once again, that's Ying Wang, Ph.D., Senior Scientist of Manufacturing Sciences at AbbVie Bioresearch Center, for sharing his knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia.com/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com, and we'll get those answers back to you. Thank you, everyone. Have a great day.